The dictionary definition of a stem cell is an unspecialized cell that gives rise to differentiated cells. Now, what does that mean? Stem cells are like normal cells, except they are undifferentiated, which means that they have no single function. Like a template or a master copy, stem cells can give rise to all the hundreds of different types of cells in the body. When a stem cell divides, it can either remain a stem cell or become specialized. They can renew themselves through cell division, even after long periods of inactivity. Hematopoietic stem cells, or HSC, are a specific type of stem cell found in bone marrow and sometimes in the blood. They are responsible for the constant production of billions of blood cells each day and can differentiate into all types of specialized blood cells. Over the course of 50 years, we have developed sufficient understanding of HSC to actually utilize them. HSC are prized for their versatility and unparalleled ability for self-renewal.
There are three main types of stem cells, which scientists can use for medical treatment: somatic or adult stem cells, pluripotent or embryonic stem cells, and induced pluripotent stem cells.
There are a few adult stem cells in your body's tissues. These stem cells replace existing, worn-out cells in your tissues, but can only differentiate to produce the specialized cell types of the tissues. Embryonic stem cells are from leftover embryos, usually 4 to 5 day old blastocysts. They can possibly develop into many more types of cells, almost every type in the human body. No discussion of stem cells would be complete without mention of the ethical debate surrounding them. In 1998, scientists discovered how to remove stem cells from human embryos. The resulting embryonic stem cells have greater differentiation potential and can divide much more rapidly and readily. They can be grown more easily in the laboratory, meaning a larger supply of higher quality stem cells.
Unfortunately, extracting stem cells from an embryo destroys it. Most of the embryos involved in stem cell research are left over from in vitro fertilization and almost never from aborted fetuses, as generally believed. Despite this, the ethical controversy of destroying embryos in order to extract stem cells persists. This may be resolved through increased use of induced pluripotent stem cells. iPSCs, or induced pluripotent stem cells, are normal differentiated adult cells that have been de-differentiated, or genetically reprogrammed to an embryonic stem cell-like state. Only a few genes must be reprogrammed in an ordinary cell to become an iPSC. These genes are most commonly brought into the cell using viruses. There have been newer methods discovered, such as nuclear programming. In addition, recent research on mice has indicated that immersing cells in an acidic environment can also reprogram the cells. Human stem cells have the potential to be very useful. Once we discover how they work, namely how an undifferentiated stem cell becomes a differentiated cell, we can unlock some of the secrets surrounding turning genes on and off. Human stem cells can also be used to test drugs. Most importantly, stem cells are at the center of a new branch of medicine called regenerative medicine, the process of replacing, engineering, or regenerating human cells that have become damaged, old, dead, or are malfunctioning. In the forefront are hematopoietic stem cells. Studies have shown that a single HSC transplanted into an irradiated mouse can create blood components, as well as epithelial cells and skin, all of which develop from different layers in the embryo. HSC are already being used to treat patients with cancer, blood disorders, and immune disorders. One example is bone marrow transplantation, a critical stem cell-based therapy. The transplanted bone marrow helps restore stem cells destroyed by chemotherapy. One day, various types of stem cells may be used to create internal blood vessels, hearts, even neurons.
Stem cells connect to the Piglo lab in that they both concern expression of genetic material. All human cells contain the genetic material to become any type of cell. However, as you develop, irrelevant genetic material is turned off. Each normal adult cell expresses only a fraction of its genes. In stem cells, everything is still capable of being expressed. A great advancement in the field of stem cells would be to figure out an easy and cheap way to reverse engineer this process, in essence to create induced pluripotent cells. In order to express genes, you must transcribe DNA into mRNA using RNA polymerase, then translate mRNA into a string of amino acids, forming a polypeptide which will fold into a protein. In prokaryotic cells such as the bacteria used in the pea-glow lab, there are two ways of controlling gene expression, positive and negative control, both of which cut off the DNA to RNA to protein process at the transcription stage. This turning on and off of genes is also present in the pea-glow lab. Namely, the green fluorescent protein gene is only expressed in the presence of arabinose, because in the presence of arabinose, the green fluorescent protein gene is able to be transcribed.